Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack series.
It is wonderful to be back with you.
Today we are exploring the biological machinery of the mind, from the microscopic switches that allow neurons to talk to the environment that rewrites our genetic code.
It really is a journey, isn't it? We're starting at the atomic level. We're literally going to be looking at individual ions and the shape of proteins.
And then zooming all the way out.
All the way out to the systemic level. I mean, we'll be talking about how the bacteria in your gut might be, whispering to your brain. It's a huge scope.
And I want to be clear, this isn't just a Biology 101 review for everyone. We are really trying to get at the mission here.
Right.
Which is to understand not just what makes the brain work, but how it physically moves, changes, and adapts. We're looking at the hardware.
The receptors.
The software, which would be the neurotransmitters. And then this third layer, which I find fascinating, the user settings.
Epigenetics. Yeah, that's a perfect framework. Because, for so long, neuroscience was it was very focused on the big stuff, the anatomy.
Like a map of a country.
Exactly.
Yeah.
This is the frontal lobe, this is the cerebellum. But the real magic, the actual, the processing of reality that happens at a scale you can't see on any map, it happens at the synapse.
So, okay, let's start there. Let's build our foundation with what we think we know. And I want to start with something that isn't there.
Something that isn't there.
The silence. The physical gap. I think, for me anyway, the biggest misconception was that the brain is like a circuit board in my computer.
Hardwired.
Yeah, wires soldered directly to other wires. You'd expect this continuous flow of electricity.
And that assumption is, well, it's the most important wrong assumption you can make. It's the design feature that makes us who we are. Neurons, the cells doing the talking, they do not touch.
Not at all.
Physically, they do not touch each other. There is a gap.
A tiny little canyon.
A tiny physical chasm called the synapse. And this gap is everything. Because if an electrical signal is flying down one neuron and it's traveling at, hundreds of miles an hour,
It's a wall.
it hits a dead end. It can't just jump across like a spark.
Yeah.
The electricity stops, the signal dies right there at the edge of the cell.
So to cross that canyon, it has to change form.
It has to be converted. The information has to go from being electrical, a voltage, to being chemical.
A chemical handshake.
That's it exactly. The first neuron releases a little cloud of chemicals, neurotransmitters, that float across that gap to the other side. And this is why we're not robots.
What do you mean?
Well, if we were hardwired, then input A would lead to output B every single time. No variation. But because there's a gap, and because it relies on chemistry,
The signal can be changed.
it can be tweaked, it can be amplified, it can be dampened, or the other side can just ignore it completely.
So the synapse is where all the negotiation happens.
Oh, that's a beautiful way to put it. Yes. The synapse is the negotiating table of the mind.
So let's meet the negotiators then. The chemicals themselves. The sources we have here, they kind of group them into what I'd call the big players. But it's not just a random soup of chemicals, is it? There's a structure to it.
Oh, absolutely. There's a hierarchy. If you want to stick with a car analogy, which actually works surprisingly well for this,
What?
you have your pedals and then you have all your dashboard settings. The pedals are glutamate and GABA.
All right, let's start with the gas pedal. Glutamate.
Glutamate is the primary excitatory neurotransmitter. It is the absolute workhorse of the brain.
Excitatory meaning it makes things happen.
It means go. In the mammalian brain, if one neuron needs to tell another neuron to fire, do something, pass this message along, it uses glutamate.
Is it like a specialized chemical for certain thoughts?
No, not at all. It's ubiquitous. It is absolutely everywhere. If you didn't have glutamate, your brain would just turn off. It provides the go signal for almost everything. Moving your finger, solving a math problem, remembering a face.
But you can't just drive a car that only has a gas pedal. That sounds like a disaster movie waiting to happen.
Chuckles. It would be a very short movie, yes. You absolutely need a break. And that is GABA.
GABA.
Gamma-aminobutyric acid. GABA is the primary inhibitory transmitter. So when GABA is released, it floats across and tells the next neuron, hey, calm down, don't fire. The signal stops here.
And this isn't just about stopping a thought in its tracks. The sources seem to imply it's more about precision.
It's all about precision. Think about tuning an old radio. You don't just crank up the volume to hear the music, right?
No, you'd get deafened by the static.
Exactly. You turn up the volume, that's glutamate, but you also have to carefully turn down the static. That's GABA. Inhibition is what sculpts the signal. It quiets down all the background noise so the really important message can stand out.
And the stakes for this balance are pretty high, aren't they? It's not just about a fuzzy radio signal.
The stakes are life and death, second by second. The balance between glutamate and GABA is quite literally the balance between a seizure and a coma. If you have too much gas, too much glutamate, and not enough brake, the brain goes into this runaway positive feedback loop of excitation. That's a seizure.
And the other way.
Too much brake, too much GABA, and not enough gas. The whole system just shuts down. That's unconsciousness. Or a coma.
Yeah.
You need both pedals working in this terrifyingly delicate balance just to be sitting here having this conversation.
That is both comforting and terrifying at the same time. Okay, so those are the pedals. But then we have the ones people have actually heard of. The celebrities.
Ah, yes.
Dopamine, serotonin. Everyone's talking about getting their dopamine hit. Where do they fit in if they're not the gas or the brake?
They're what we call modulators. So in our car, they aren't the pedals driving the car forward or stopping it. They're more like the climate control,
Right.
or the suspension settings, or the GPS navigation.
They change the quality of the ride.
That is, and exactly, they change the state of the system. Take dopamine. It's famous for reward, motivation, movement, but it doesn't usually cause a neuron to fire all by itself.
But what does it do?
It changes how sensitive that neuron is to glutamate. It might get a little whisper of dopamine that says, hey, that next glutamate signal that comes in, pay extra close attention to that one because that one is associated with pizza.
Chuckles, or something a little more evolutionarily significant, I hope.
Well, pizza's pretty significant if you're hungry enough. But yes, food, reproduction, spotting a predator, and serotonin does something similar for mood and sleep and appetite. Acetylcholine is critical for attention and for contracting your muscles, but they're all modulators. They're turning the knobs on the dashboard to fine-tune the system's response, the main drivers.
So we've got these keys and neurotransmitters floating around. We have the gas, the brake, the climate control. But a key is totally useless if you don't have a lock. And this brings us to the hardware, the receptors.
Yes, the receptors. These are the protein machines sitting on the surface of that receiving neuron, waiting for the keys. And this is where the sources, they get really granular in a way that is just fascinating, because the word receptor is way too broad.
Like vehicle.
Exactly like vehicle. A bicycle and a tank are both vehicles, but they work very, very differently.
And in the brain, we basically have two families of receptors, two really different kinds of locks, ionotropic and metabotropic. Now, I want to spend a little time here, because this difference seems to be the fundamental split in how our brain processes time.
That is a very sharp way to put it. Let's start with the ionotropic receptors. These are your fast receptors.
And when we say fast, what are we talking about?
Millisecond time scales, basically instantaneous. For all intents and purposes, it's immediate. They're also called ligand-gated ion channels.
Okay, let's break down that jargon. Ligand-gated.
A ligand is just the scientific name for any molecule that binds to a receptor. So in our case, it's the neurotransmitter.
The key.
The key. And gated means there's a gate or a door. So the mechanism is beautifully simple. The neurotransmitter, the key, floats across the synapse, finds its lock, and plugs in. Boom.
Boom.
The receptor protein physically and immediately changes its shape, opening a hole right down its center, a channel.
It's a mechanical action. The key turns, the door opens.
Exactly. And because there's a different electrical charge inside the cell versus outside, charged particled ions like sodium or potassium go rushing through that open door like a crowd storming a stadium gate.
And that flood of ions is what changes the electricity of the receiving cell.
Instantly. This is absolutely essential for things like vision or hearing or pulling your hand away from a hot stove. You don't have time to form a committee and discuss it. You need the door to open now.
Right. The analogy in the notes was perfect for this. Ionotropic receptors are like a simple door handle. You turn the handle, the door opens, you walk through. It's direct, it's mechanical.
It is. Now, compare that to the other family, the metabotropic receptors. These are the slow ones.
And slow in brain time means what? A few seconds?
It could be hundreds of milliseconds, up to seconds, and the effects can last for minutes or even longer. In the world of neurons, that is an eternity.
Why are they so slow? Is it just an inefficient design?
Knuckles. No, they're not lazy. They're bureaucratic. They're complex because they do a much more complex job. They're often called G-protein coupled receptors, or GPCRs.
Okay.
Now here, the neurotransmitter binds to the receptor, But the receptor itself does not have a channel. It doesn't open a hole.
So it's a door handle that isn't connected to a door. What's the point?
It's not a handle, it's a doorbell. When the key turns in this lock, it doesn't open the door. It activates a little guy on the inside of the cell, a middleman.
A middleman.
A G protein. This middleman then splits into parts, an alpha subunit and a beta gamma subunit. And these little messengers go running off inside the cell to trigger a whole cascade of events.
A chain reaction.
A huge chain reaction. They might activate an enzyme like adenyl cyclase,
Which then makes another messenger, CMPP.
which then activates another enzyme, protein kinase A.
Okay, I see why it's slow. It's a game of corporate telephone.
It is. But it's a very useful game of telephone because it amplifies the signal. One single binding event on the outside can result in thousands of molecules changing inside the cell. It changes the whole state of the cell. It's modulation.
So to stick with the analogy, if the ionotropic receptor is the door handle,
The metapopic receptor is you pressing the doorbell. That rings a chime in a distant room, which wakes up the butler, who then puts on his coat, walks down the hall, checks the people, and then decides whether to open the door or maybe just turn up the thermostat instead.
right, so ionotropic is for fire now, and metabotropic is for let's change the whole mood in this room.
Precisely. And that fundamental distinction is why you can react to a loud bang instantly. That's ionotropic.
Yeah.
But a feeling, a mood like anxiety or joy, can linger for minutes or hours. That's the work of metabotropic cascades.
That makes so much sense. Now, I want to double-click on those fast ones, the ionotropic receptors. Because the architecture that the sources describe, especially in some of these reviews, it's just incredible. These aren't just, amorphous blobs of protein. They're intricate little machines.
They're masterpieces of nanoengineering. And evolution has, it seems to have settled on two primary architectural designs for these fast channels.
The superfamilies.
Yes. The first one is the cis-loop superfamily.
Cisloop. Sounds like a brand of breakfast cereal.
Chuckles. It actually refers to a very specific structural signature. There's a loop of 13 amino acids in the protein that's held together by a disulfide bridge, which involves two cysteine residues, so cis-loop.
And what kind of receptors belong to this family?
It's a big and important one. It includes the nicotinic acetylcholine receptor. That's what nicotine from cigarettes binds to. It includes one of the serotonin receptors, the 5-HT3 type. And crucially, it includes the main inhibitory receptors.
The brake pedals.
The brake pedals. GABA-O and glycine receptors are both cis-loop receptors.
So what do they look like?
I want you to picture 5 people standing in a circle holding hands.
Okay, 5 people in a ring.
Each person is one subunit, a single protein chain. All 5 together form the complete receptor. The space in the middle of their circle right between their feet, that's the ion channel, the pore. We call this a pentamer. 5 parts.
That's a 5-sided donut.
A 5-sided protein donut, yes. Now, the inner wall of that hole is lined by a specific part of each of those 5 proteins. Let's call it their right leg. In biology, it's the M2 transmembrane helix.
And when the channel is closed,
Those 5 legs are all bunched up in the middle, blocking the path. The circle is tight. Nothing can get through.
and then the neurotransmitter binds.
It binds up near the top, near the shoulders of these proteins. And that binding causes a physical shift. The whole structure twists or tilts, and in doing so, the proteins pull their legs back. The circle widens, the pore opens.
And the ions flood through. It's a machine made of meat.
It is pure nanomechanics. And then you have the other major family, the ionotropic glutamate receptors, or IGLURs. These are the ones that respond to our gas pedal, glutamate.
Are they also 5 people holding hands?
No, they are tetramers, 4 subunits.
4 people.
4 people. But their structure it's much taller and arguably more complex. The source material describes it as a layer cake design.
A layer cake.
Imagine a 3-tiered wedding cake. The bottom tier is embedded in the cell membrane, that's the channel itself. The middle tier is what we call the agonist binding domain. And the top tier, which sticks way out into the synapse, is the amino terminal domain.
Okay, and this is where that clamshell mechanism comes in. This was the visual that really made it click for me.
It's one of the most beautiful discoveries in structural biology. That middle tier of the cake, the part where glutamate actually binds, it's shaped like a Pac-Man mouth or a clamshell.
And it just sits there open.
It sits there open, waiting. When a glutamate molecule happens to wander into that mouth, the clamshell snaps shut on it.
It bites it.
It literally bites down and traps it. Now imagine that the bottom jaw of that clamshell is connected to a rope, and that rope goes down and is tied to a little trapdoor in the floor, or the channel gate.
Okay.
When that mouth snaps shut, It physically pulls on the rope. It creates tension.
And it yanks the trapdoor open.
It yanks the gate open. That physical closing motion of the clamshell is what directly drives the opening of the ion channel at the bottom.
So it bites the neurotransmitter, and the force of that bite is what opens the gate.
That's a very vivid way to put it, but fundamentally, yes. It pulls on these little linker segments that connect the binding domain to the transmembrane domain. It's an incredible piece of machinery.
So we have the software, the neurotransmitters, we have the hardware, these amazing receptor machines. But the outline also mentions the user settings, epigenetics. Now, I feel like this word gets thrown around a lot in pop science. Sometimes it feels like it just means magic.
Chuckles, it can feel that way. But the sources we have really define it mechanically. Epigenetics literally means above genetics. And the core concept is your DNA is not your destiny. It's a script.
But you can edit the script.
You can decide which scenes to shoot and which to leave on the cutting room floor. The classic visualization is Waddington's landscape.
Right, the marble on the hill.
Exactly. Imagine a marble at the top of a hill, and the hill has all these valleys and ridges carved into it. As the marble rolls down, it has to choose a path. And once it rolls down into one valley, it's very hard for it to get back up over the ridge and into a different one.
And the marble is a cell.
The marble is a stem cell, and the valleys are its possible fates. Becoming a neuron, a skin cell, a liver cell. Epigenetics is what carves those valleys into the landscape.
But how? Mechanically, how does a cell know to ignore a huge chunk of its own DNA?
There are two main mechanisms highlighted in the texts. The first is DNA methylation. This is like putting a tiny chemical padlock on a gene.
A padlock.
A team of enzymes adds a methyl group, a carbon, and 3 hydrogens onto the DNA itself, specifically at cytosine bases. And this tag acts as a gatekeeper. It generally silences the gene. It makes it inaccessible to the machinery that reads the DNA.
So methylation is basically an off switch.
In many contexts, yes. The second big mechanism is histone modification. You have to remember, your DNA isn't just a loose strand floating around. It's 6 feet long, crammed into a microscopic nucleus.
So it has to be packaged.
It's wrapped around these protein spools called histones, like thread on a spool. And that whole complex is called chromatin.
Okay.
Now, if that thread is wound super tight around the spools, you can't read the instructions written on it. If it's loose, you can. Histone modification is the process of adding other chemical tags, like acetyl groups, to the tails of these histone spools.
And that changes how tight the wrapping is.
Exactly. Acetylation, for instance, usually neutralizes a positive charge on the histone tail, which makes it let go of the negatively charged DNA. It loosens the coil.
It opens the book to that chapter so the cell can read it.
That's the perfect analogy. So you have DNA as the library of books, and epigenetics is the librarian going around and putting padlocks on some books while highlighting and opening up others.
So that's the foundation, the chemical handshake, the pentamers and tetramers, the genetic volume knobs. But here's where it gets really fun, because we actually look at the data, the real science. It's not nearly that simple. There are huge active debates.
Science is never settled, especially in neurobiology. I mean, you have to appreciate we are trying to take pictures of individual protein molecules that are moving at the speed of heat while they're embedded in a greasy fluid membrane. It is an astonishingly difficult thing to do.
Let's start with one of those debates, the mechanics of gating. We said the door opens, but the sources suggest there is a massive scientific fight over how the door opens, specifically with those cis loop receptors, are 5 people in a circle.
Right, the conformational wave debate. Think about it. The neurotransmitter binds way up at the top of the receptor at the head and shoulder area. The gate, the part that actually opens and closes, is way down at the feet. That's a distance of about 60 angstroms.
Which is tiny for us, but for a molecule, that's a huge distance.
It's a vast distance. So how does the information that a key has landed in the lock travel all that way down to the door? It's not telepathy. It has to be a physical wave of movement that propagates through the protein.
Like cracking a whip, the motion travels down the length.
A very similar idea. Now, for the cis loop family, we know a key part of the binding is that a loop of protein, loop C, actually moves to cover the neurotransmitter like a lid. It's a capping motion. It traps the molecule in place.
Okay, so step 1 is clear. Capture the key.
Right. But then what happens? Does the channel, those 5 legs at the bottom, does it twist open? Or does it tilt?
Twist or tilt. This sounds like a dance move debate.
Right.
Why does it matter so much?
It sounds trivial, but from a drug design perspective, it changes everything. The twist model suggests that the 5M2 helix lining the pore rotate. kind of like the aperture on a camera lens. You know that opening sequence in James Bond movies?
The gun barrel, right, the iris opening.
That's the twist model. But the tilt model, which is supported by some really clean structures of bacterial versions of these channels, suggests the whole wall of the channel simply collapses or tilts outward, widening the gap, more like a flower blooming.
And we really don't know for sure which one it is for our own receptors.
We have evidence for both. Different receptors might do it differently. And that means if you're a pharmacologist trying to design a drug to, say, jam the door open or wedge it shut, you need to know if you should be building a wedge for a twisting door or for a tilting door. They're different mechanical problems.
And what about for the glutamate receptors, the clamshells? There was a concept there that seemed really counterintuitive. Desensitization?
Ah, yes. The tension mechanism. Remember our rope pulling on the trap door?
Yeah, the clamshell bites, pulls the rope, the door opens. Simple.
Right. But now imagine the clamshell stays shut. It's holding on tight to that glutamate molecule. The rope is taut. The trap door is open. But if it stays like that for too long, the rest of the protein structure down at the bottom basically says, I can't maintain this tension anymore. And the linkers, the ropes, they lax, they go slack.
Even though the clamshell is still biting down.
Even though the clamshell is still closed tight, the linkers give up, they lose their connection to the gate, and the trapdoor slams shut again.
So the key is still in the lock, turned to the open position, but the door just slams shut anyway.
That is desensitization, and it is a critical safety mechanism. If that channel stayed open indefinitely, ions, especially calcium, would just flood into the cell, building up to toxic levels. It would kill the neuron.
That's called excitotoxicity.
Exactly. So the receptor has this built-in circuit breaker and says, okay, we've been open long enough, and it shuts itself off to save the cell even if the go signal is still there.
He has a built-in attention span.
That's a great way to think about it. And understanding that slip in the linkage is key to understanding why some drugs can lose their effectiveness over time.
This is just incredible engineering. Speaking of which, we had to talk about the superstar of complex engineering, the NMDA receptor. The outline labels this one as the coincidence detector. And this stood out to me as the most philosophical piece of biology in the whole stack.
It absolutely is. The NMDA receptor it's a unique beast among all the receptors we've discussed. It is high maintenance, it's finicky, it requires not one, but two different keys to even think about opening.
Two keys, like a safe deposit box or a nuclear launch sequence.
Very much like that. First, it needs glutamate, just like the other iglors, but that's not enough. It also needs a coagonist, which is usually the amino acid glycine or another one called D-serine.
Okay, so you need key 1 and key 2 turned simultaneously.
But wait, there's more. Even with both keys in their locks, the door still will not open.
Why not? Is it jammed?
It is blocked. There is a bouncer standing in the doorway, a single magnesium ion.
Just one ion of magnesium.
An Mg2 plus ion. At the normal resting voltage of a neuron, when the cell is just sitting there, quiet, the inside of the cell is negatively charged. The magnesium ion is positively charged.
Opposites attract.
Opposites attract. So the magnesium is pulled, it's sucked deep into the channel pore, and it just gets stuck there. It sits in a binding site inside the channel and physically plugs the hole like a cork in a bottle.
So nothing can get through. It's corked shut.
Completely corked. Glutamate can be there, glycine can be there, the clamshells can bite down, but as long as that magnesium bouncer is in the doorway, no ions can flow.
So how on earth do you get rid of the bouncer?
You have to change the electricity, you have to pay them off with voltage, the neuron has to be depolarized.
Meaning it has to become less negative?
It has to become positively charged on the inside. If the inside of the cell becomes positive, it will now repel the positively charged magnesium ion, like trying to push two north poles of a magnet together. It physically boots the bouncer out of the channel.
Okay, let me try and put this all together. For an NMDA receptor to finally open, you need the sending cell to be talking.
Releasing glutamate, yes.
You need the coagonist, glycine, to be present.
Correct.
And you need the receiving cell to already be active and excited from other inputs, enough to change its voltage and kick out the magnesium.
That's it. It detects the coincidence of presynaptic activity, the release of glutamate, and postsynaptic activity, the depolarization. It will only open when both sides of the synaptic conversation are shouting yes at the exact same moment.
Cells that fire together wire together.
That is the literal molecular definition of Hebbian learning. Because when that magnesium plug finally pops out, the ion that comes rushing in isn't just sodium, it's calcium.
And calcium is a powerful messenger.
Calcium is a signal. It goes into the cell and it screams, hey, this connection we just used, this specific synapse, it's important. It predicted something. Strengthen it. Build more receptors here. Make this connection permanent.
That is just wild. A single magnesium ion is the gatekeeper of associative memory. If that magnesium block didn't exist,
we wouldn't be able to form associations. We couldn't learn cause and effect. Every signal would just be noise. The magnesium block is what allows the brain to distinguish between a random coincidence and a meaningful correlated event that's worth remembering.
It's a tiny atomic detail that dictates our entire experience of reality. Now, another area of debate, and this one feels a bit like quantum mechanics, partial agonism.
This is a huge deal for pharmacology.
So we get it. A full agonist is a key that opens the door all the way. An antagonist is a key that breaks off in the lock and keeps it shut. But a partial agonist opens it. What, halfway?
Well, that's the multi-million-dollar question, isn't it? What does halfway even mean for a single protein molecule? And there are competing theories.
The clamshell theory again.
For the AMPA and kainate type of glutamate receptors, yes. One theory is that a partial agonist causes the clamshell to close only partially. It takes a smaller bite, a nibble.
So the rope isn't pulled as tightly?
Exactly. The tension on the linkers is less, and so the channel gate only opens to a subconductance level. It is, quite literally, a half-open door that lets fewer ions through per second.
Okay, that's pretty intuitive.
But for NMDA receptors, some of the data suggests something different. It suggests the clamshell closes fully, just like with a full agonist, but that the closed state is unstable. It's jittery. It flickers open and closed very rapidly.
So it's not a half-open door, it's more like a flickering light bulb.
Precisely. The average current flowing through is lower, not because the opening is smaller, but because the door is slamming open and shut 1,000 times a second instead of staying steadily open.
Why does this distinction even matter? As long as less stuff gets through, who cares if the door is half open or just flickering?
The people designing our medicines care immensely. If you want to treat something like schizophrenia or depression, you probably don't want to blast a receptor fully open. That can cause all sorts of side effects. And you don't want to shut it down completely. That can be just as bad. You want a dimmer switch.
You want to modulate it.
You want to gently tune the system. And if you don't understand the physical reason why your drug is acting like a dimmer switch, whether it's by holding the door ajar or by making it flicker, you can't rationally design a better one.
That makes perfect sense. Okay, one last thing in this debate section that just blew my mind was RNA editing, the QR site. This sounded like the body is actively hacking its own source code.
Oh, this is a heavy hitter. We're all taught the central dogma in high school biology, right? DNA makes RNA, and RNA makes protein.
The blueprint makes a photocopy, and the photocopy is used by the factory to make the product.
But what if, after the photocopy is made, the cell takes out a pen and changes one of the instructions on it?
It edits the blueprint after it's been copied.
Yes. In the gene for the GluA2 subunit of the AMPA receptor, one of the 4 pillars in that layer cake structure, there's a specific spot in the DNA code that calls for the amino acid glutamine. It's represented by the letter Q. But the cell has an enzyme called ADR2 that, once the RNA copy is made, finds that one spot and chemically changes that instruction to call for arginine instead, the letter R.
So it's just one amino acid change out of hundreds?
Just one, but it changes everything about the receptor's function. Glutamine is electrically neutral, arginine is bulky and has a strong positive charge.
So what happens when you stick a big, positively charged rock in the middle of a channel that's supposed to let positive ions through?
It acts like a bouncer. It repels calcium ions. A receptor with glutamine at that spot is permeable to calcium. A receptor with arginine at that spot blocks calcium.
So this one little edit turns the calcium flow off.
It does. And in the adult human brain, nearly 100% of all the GluA2 subunits have been edited from Q to R. They are calcium impermeable.
But why? Why go to all that trouble?
To prevent excitotoxicity. The brain wants the fast signaling that comes from sodium rushing through these channels, but it doesn't want the powerful, potentially toxic signaling that comes with calcium flooding in every single time. It's a way of protecting the neuron from overstimulation.
And what happens if this editor calls in sick? If the ADR2 enzyme fails?
If that enzyme fails, you get unedited receptors with glutamine at the QR site. Calcium floods into the motor neurons, and the neurons die. This specific failure, a lack of RNA editing, is very strongly linked to the neurodegeneration seen in ALS or Lou Gehrig's disease.
That's just frightening. A single typo in the RNA editing process, or rather, a failure to correct a typo, can lead to a fatal disease.
It just highlights how incredibly dynamic this whole system is. We are constantly rewriting our own biological instructions. We're not just reading the DNA, we are actively editorializing it moment by moment.
This leads us perfectly into the next section. The frontiers. The stuff that is actively changing how we look at the brain right now. We've spent all this time talking about the receptor itself, but the research suggests we've been making a fundamental mistake by looking at the receptor in isolation.
We have been studying the soloist while completely ignoring the rest of the orchestra.
The entourage, as the notes call it.
Yes, yes. For decades, the way we studied these proteins was in a totally artificial system. We'd take the DNA for a receptor, put it into a kidney cell or a frog egg, measure the electrical currents, and think, aha, this is how it works.
But we were studying naked receptors.
In the wild, they were clothes, and they have handlers. We call them auxiliary subunits. The most famous discovery here was a family of proteins called TARPs.
TARPs.
Transmembrane AMPA receptor regulatory proteins. The first one discovered was called stargazin.
That's a great name for a protein.
It comes from the mouse it was discovered in. It was a mutant mouse that had seizures and this weird upward head-tossing movement. It looked like it was gazing at the stars.
And it turned out the mouse just had a broken protein.
It was missing this stargazin protein, and what they found was that stargazin acts like a personal chaperone for the AMPA receptor. It grabs onto it inside the cell, guides it to the synapse, staples it to the membrane so it doesn't float away, and even changes how the channel opens and closes.
So without stargazin, the receptor is just lost.
It might never even make it to the cell surface, or if it does, it doesn't get anchored at the synapse and doesn't work correctly. It creates what we call silent synapses.
This just adds another whole layer of complexity. It's not just do you have the gene for the receptor, it's do you have the gene for the chaperone that gets the receptor to the party and makes sure it behaves.
Precisely. And stargazin is just the tip of the iceberg. We're now discovering whole families of these auxiliary proteins, cornichons, CCAMPs, GSG1L. It's a whole zoo of molecules that hug the receptor and tune its function in very specific ways.
It sounds like those user settings we talked about, epigenetics, are getting more and more physical. It's not just chemical tags on DNA anymore, it's physical clamps on the proteins themselves.
Well, speaking of those user settings, we have to talk about the biggest frontier in the study of memory right now, neuroepigenetics.
We touched on epigenetics in the intro, the marble rolling down the hill to decide a cell's fate. But the source material suggests this isn't just for developing babies, it's how we are storing our memories right now.
This is a complete paradigm shift. The old model was simple. A synapse gets stronger, a memory is formed. Done. But there's a problem with that. Proteins don't last forever. The very receptors we've just been talking about, they degrade. They're broken down and replaced every few days.
So wait, if the physical hardware of the synapse is being completely swapped out every few days, how do I remember my 5th birthday party from decades ago?
Exactly. How can a memory persist for a lifetime if the molecules that supposedly store it only last for a weekend? The theory is that the memory isn't just stored in the proteins, it's burned into the genome's expression pattern.
Back to the library.
A learning event triggers that calcium influx through the NMDA receptor we discussed. That calcium signal travels all the way to the nucleus and activates enzymes that modify the histones or methylate the DNA at specific genes related to memory and plasticity.
So long-term memory is literally an epigenetic scar on your DNA.
A mark, yes.
Yeah.
It's like opening the book to a specific page and putting a permanent bookmark there that says, keep this chapter open. Keep making these specific proteins to maintain this specific synapse.
This raises a massive and slightly scary question. Unlike genetic mutations, which are permanent damage, you said epigenetic marks are potentially reversible.
Theoretically, yes. They are enzymatic processes.
So can we erase trauma?
That is the billion-dollar question. If we can identify the specific enzyme that wrote the traumatic memory, the writer that put that bookmark there, could we develop a drug that inhibits that enzyme and allows us to erase the emotional weight of that memory?
It sounds like eternal sunshine of the spotless mind.
But with targeted pharmacology. And the flip side is just as profound. In aging, our brains become less plastic. Could we use drugs like HDAC inhibitors to restore the plasticity of a younger brain by chemically forcing those epigenetic books to open back up?
That is both incredibly hopeful and slightly dystopian. But we can't talk about frontiers without talking about the other alien intelligence living inside our body, the microbiota gut-brain axis.
For 100 years, we thought the brain was a fortress, protected by the blood-brain barrier. It kept everything out. But now we know that the tenants living in the basement, the trillions of bacteria in your gut, are constantly talking to the penthouse.
And they're very loud tenants.
They are. The gut microbiota produce metabolites, like short-chain fatty acids that get into our bloodstream. They produce neurotransmitters. Did you know that a huge percentage of the body's serotonin is actually produced by cells in the gut, influenced by bacteria?
I have heard that. But does it actually get to the brain? I thought serotonin couldn't cross the blood-brain barrier.
The serotonin itself might not cross easily, but its precursors, like tryptophan, do. And more importantly, the signals travel up the vagus nerve, a direct highway from the gut to the brain. The sources highlight the studies on germ-free mice.
Mice raised in a sterile bubble with no gut bacteria at all.
Yes. And what happens to their brains? Their development is altered, their neuroplasticity is different, their expression of NMDA receptors is different.
Hold on. The bacteria, or lack thereof, in the gut changes the number of NMDA receptors in the brain.
Yes. It proves that a healthy microbiome is an essential environmental factor for building a normal brain architecture.
So this idea of psychobiotics, it's a real potential future, taking a specialized probiotic pill to treat anxiety or depression.
It is being researched very heavily. Now, we have to be cautious. Mice are not men. The human system is vastly more complex. But the link is undeniable. Your diet affects your gut bacteria, which affects your epigenetic settings, which affects your receptor expression, which ultimately affects your mood. It's all one interconnected system.
And finally, under frontiers, we have the technology that's making all of these discoveries possible. Structural biology.
Cryo-EM, cryo-electron microscopy.
We've been using analogies like taking a picture of these receptors, but this technology is next level.
It's a revolution. For decades, the only way to see a protein structure was X-ray crystallography, which meant you had to trick billions of protein molecules into lining up in a perfect, solid crystal.
Like freezing a bird into a block of ice to see what it looks like.
It's a great analogy. It distorts the bird. And these receptors are floppy, greasy things embedded in a membrane. They hate forming crystals. Cryo-EM changed the game. Now we can just take them, flash freeze them in a thin layer of ice vitrification, and take pictures of them in their natural state.
So we're freezing them in mid-action.
We can freeze them and see them in the open state, the closed state, the desensitized state. We can literally see the kink in the M2 helix that blocks the channel and how it straightens out when it opens. We're not just guessing about the twist versus tilt debate anymore. We are starting to watch the movie frame by frame.
That's incredible. It transforms this part of biology from a kind of guessing game into a true engineering discipline.
It does. And that brings us to the real-world impact, the implications. Why does all this insane molecular detail matter to the person listening right now? Because when these tiny machines break, or when we deliberately interfere with them, the consequences for individuals and society are profound. Let's talk about neurotoxicity.
The sources immediately pointed to organophosphates, pesticides. This is stuff that's in our agricultural system right now.
Yes, and understanding the synapse explains exactly why they are so dangerous. These chemicals work by inhibiting an enzyme called acetylcholinesterase.
Acetylcholinesterase? That sounds like the cleanup crew.
It's the janitor of the neuromuscular junction. Remember, in a synapse, you release the neurotransmitter, in this case, acetylcholine. It binds to its receptor, and the muscle contracts. But you don't want the muscle to stay contracted forever. You need to relax.
So you have to get rid of the acetylcholine.
Exactly. The janitor, acetylcholinesterase, comes in and rapidly breaks down the acetylcholine, cleaning it out of the synapse. Organophosphate pesticides bind to that janitor and kill him.
So the trash just keeps piling up.
The acetylcholine just builds and builds and builds in the synapse. It keeps banging on the door of the receptor over and over. The receptor gets massively overstimulated. The muscle locks up in a state of tetany.
Paralysis.
A rigid paralysis. And if that muscle happens to be your diaphragm, which you need in order to breathe, you suffocate. This is how chemical nerve agents work. It's how many common pesticides work on insects, and in high enough doses on us.
And it's not just about acute high-dose poisoning. The sources talk a lot about developmental toxicity.
And this connects right back to the scaffolding we talked about earlier. The developing brain of a fetus is exquisitely vulnerable. The placenta is not a perfect barrier. Things like lead, mercury, and certain pesticides can cross it and disrupt the formation of these delicate synaptic circuits.
If you build the house with crooked framing in the beginning, The windows are never going to open and close properly later on.
That is the perfect analogy. If you mess up the layout of the receptors or the epigenetic settings during that critical first trimester, you can't just go back and fix it later. This is why environmental regulations are not just about protecting nature. They are fundamental public health treaties.
Let's shift from external threats to internal diseases. Parkinson's disease. Most of us think of this as a shaking disease, the tremors.
That is the most visible motor symptom, yes. And it's caused by the progressive death of dopamine-producing neurons in a part of the brain called the substantia nigra. But the research here really highlights the non-motor symptoms.
Right, the depression, the anxiety, the cognitive decline.
These often appear years, sometimes a decade, before the first tremor. And looking at the receptor pathways helps explain why. The pathology involves these clumps of a protein called alpha-synuclein, forming what are called Lewy bodies. And these clumps don't just stay in the movement centers.
They spread through the brain.
They spread. And the sources point out that dopamine isn't just one system. There isn't just the dopamine system. There are distinct pathways. The nigrostriatal pathway controls movement, but the mesolimbic pathway is all about reward, motivation, and emotion.
So the disease hits the brain's reward and mood circuits too.
Yes. And that helps explain the profound depression and apathy that are often the first signs of the disease. The treatments we have, like L-Dopa, they're basically just dumping more raw materials into the brain to force the remaining neurons to make more dopamine. But that doesn't stop the neurons from continuing to die. It's a patch, not a cure.
Which brings us directly to that mesolimbic pathway, one of the biggest societal issues we face, addiction.
This is a case of the mesolimbic pathway being hijacked.
We talked about dopamine being that climate control system that tells your brain to pay attention to important things like food or survival. But drugs of abuse, they hack that system.
They do more than hack it. Drugs of abuse, opioids, stimulants, alcohol, they trigger a release of dopamine in the reward circuit that is bigger, faster, and more reliable than any natural reward ever could be. It's what we call an evolutionary superstimulus. It shouts at the dopamine receptors.
And the brain, being wonderfully adaptable, reacts to being shouted at.
It reacts by putting in earplugs. That process is called downregulation. The brain's control systems say, whoa, the signal is way too loud, we're going to get damaged. So it literally pulls the dopamine receptors off the surface of the cell and internalizes them. It reduces the receptor count.
So now you have fewer ears listening for the signal.
Exactly, and that works in a way to protect the cell when the drug is present. But what happens when the drug wears off? You are left with a brain that has half the normal number of dopamine receptors.
So normal life, a hug, a good meal, watching a sunset, none of it registers anymore.
It's too quiet. The natural dopamine signals are too weak to be heard by the reduced number of receptors. That is the biological basis of anhedonia. The inability to feel pleasure from normal life. And at that point, the user needs the drug not to get high anymore, but just to feel normal, just to get the volume of life back up to baseline.
And the scariest part from the notes is the connection back to epigenetics. We talked about a long-term memory being an epigenetic scar. Is addiction a scar too?
The evidence is mounting that it is. The repeated massive surges of drug-induced signaling can trigger long-term epigenetic changes in the reward circuits. The memory of the addiction gets written onto the chromatin.
And that would explain relapse.
It would explain why the risk of relapse can persist for years or even a lifetime after someone gets clean. The drug is gone. The receptors may have returned to normal levels, but the epigenetic landscape of those neurons has been fundamentally altered. The pathway is etched there, just waiting to be reactivated.
That's incredibly heavy. But let's try to end on a note of scientific optimism. We talked about the dimmer switch in pharmacology. This seems to be the way out of the sledgehammer era of psychiatric drugs.
Absolutely. This is the promise of allosteric modulation.
Can you break that down? Allosteric?
Allo means other, and steric refers to shape or space. Most of our current drugs are orthosteric. They compete for the main keyhole where the natural neurotransmitter binds.
It's a direct fight, me versus the neurotransmitter.
Right. And that usually means you either turn the receptor fully on, like a full agonist, or fully off, like an antagonist. It's a binary switch. But allosteric modulators don't bind there. They bind to a completely different spot on the receptor, a side door, or a spot on the leg of the protein.
And what do they do from all the way over there?
They just gently tweak the receptor's response. They change its shape slightly so that when the natural key comes along, it works a little better or a little worse. They're not the main actor, they're the supporting cast.
Can you give us an example?
The classic example is benzodiazepines, like Valium or Xanax. They don't open the GABA channel on their own. If you
Yeah.
put Valium on a GABA receptor and there's no GABA around, absolutely nothing happens.
It doesn't open the door by itself.
No. But when GABA is present, the Valium makes the channel open more frequently and stay open a little longer. It enhances the natural brake signal. It makes the brake pedal more sensitive, but it doesn't slam it to the floor.
And the zinc inhibition of NMDA receptors we mentioned earlier.
Zinc is a negative allosteric modulator. It binds to a separate site and turns the volume down on the NMDA receptor's response. The great hope is that we can design drugs that are exquisitely specific, that only bind to the allosteric site on one specific receptor subtype-like. Only the GLO-N to be containing receptors involved in a particular disease without messing with all the other subtypes throughout the brain.
So it's precision medicine at the atomic level.
Yeah.
Instead of systemically flooding the brain, we're trying to tune one specific string on one specific violin.
That is a beautiful image, and that is absolutely the goal of modern neuropharmacology.
We have covered a truly massive amount of ground today. We've gone from the cis loop pentamers, those 5 proteins holding hands, to the epigenetic scars of memory, to the bacterial tenants in our gut, and finally to the ethics of how we treat our own brains.
It really does reinforce the central theme, which is that the brain is plastic at every single level. From the subtle wiggle of a receptor loop to the addition of a methyl group on our DNA to the food we eat that changes our gut, we are constantly being rewritten. We are not static hardware. We are a living, breathing work in progress.
And that, I think, is the most empowering takeaway of all. We are built to change.
You're built to learn.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content, and support us on Patreon. Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.